Hello class, so we're going to continue with chapter 23, part 1. We left off on slide 68 and I realized this slide shouldn't even be in this PowerPoint. So please don't pay attention to this. We will talk about the adaptive defenses within our immune system in Anatomy 3. So I'll get this rid of this slide and repost it for you. So let's continue on with our stomach. We're going to have some peristaltic waves within our stomach. First step in this will be propulsion, where we have the waves moving from our fundus. Remember, our fundus is when we draw a line across from that esophagus entering the stomach, and it's that dome-shaped structure here. The second part of this is going to be the grinding process. So it's the most vigorous, as you can imagine, grinding the food up, vigorous part of peristalsis. So we're having the muscularis externa muscles. Remember, there's 3 layers here in the stomach working to contract and churn the chyme in the stomach. And the pyloric end of the stomach is going to act more as a pump. It's going to deliver small amounts of this chyme into the duodenum, which is the first part of the small intestines. And the third part of this is going to be retropulsion. Remember, retro means backwards. So here, when we have our peristaltic waves causing the pyloric sphincter or the pyloric valve to close up, that forces contents back into the stomach because we're trying to regulate how much is moving into the small intestines. We just want a little bit at a time. So next we're going to talk about regulation of this movement in the stomach and also the emptying that it does. So the way we regulate this is actually through the duodenum. Our duodenum will see how much chyme is moving into it and then tell its duodenal receptors and chemical receptors what to do. So here's our chemoreceptors. Again, these are just chemical receptors, and our stretch receptors. That's going to trigger our enterogastric reflex. And the chyme moving into our duodenum will also cause our enteroendocrine cells to secrete our enterogastrones. This is going to be like secretin, and secretin is going to help us to inhibit the gastric gland secretion and gastric motility. It'll also increase our output of the pancreatic juices and will increase our bile output. Another type of enterogastrone is going to be our cholecystokinin, or CCK. And our cholecystokinin, that is going to inhibit our stomach's secretory activity. It's going to increase the output of pancreatic juices, expel our stored bile in the gallbladder, and relax our sphincters so that we can let the bile and pancreatic juices move into the duodenum from the duct system. All right. What the reflex and these enterogastrones are going to result in is going to be a decreased contractile force of the stomach and decreased rate of emptying. So basically, we're using this more as our storage. We're keeping our chyme within our stomach until our duodenum is ready for it to move back in. Now, our stomach is going to empty in approximately 4 hours. Sometimes it's a little faster than that. It could be as fast as 2 hours. But when we have an increase in fatty chyme, then it's going to increase our time to 6 hours or more. And our carbohydrate-rich chyme is going to move quickly through the duodenum. Now let's move into our major accessory organs of our digestive system. So we have our liver, gallbladder, and pancreas, and let's just review where these are. We have our liver in our upper right quadrant. Gallbladder is going to be in the inferior surface of the liver, and then our pancreas is found posterior to our stomach. Remember, our pancreas is a retroperitoneal structure. What does our liver do? Well, it does a ton of things, but main function is going to be a digestive function and produce bile. Bile is going to help to emulsify fat. Our gallbladder, because it has that word bladder in there, it's going to help in storage. And gall is referring to bile, so we just store bile. And our pancreas is going to supply most of the enzymes that are needed to digest the chyme, and it'll secrete it into the duodenum here, as well as using the bicarbonate in it to neutralize the stomach acid. Remember, we want acidic environment in the stomach, basic environment in the duodenum. All right, we'll start off with the liver first. This is our largest gland in the body. It's going to weigh close to 3 pounds, and we have a couple different lobes here. We have our right lobe, which is larger, the left lobe, which is smaller, and then when you look at the inferior view of the liver, you will find the caudate lobe, which is more posterior, inferior and posterior, I should say, and that makes sense because our caudate term means tail, so you find the tail in the back. And then you have your quadrate lobe here. Quadrate because we have this square shaped lobe here. And I always like to think Q and A. So Q for quadrate, A for anterior. And then we have our falciform ligament. You can see that falciform ligament it's sitting in between your right and left lobes here. And it'll basically connect the liver to the anterior abdominal wall. We'll also have part of the diaphragm up in this region. And we also have our round ligament here. This is also known as your ligamentum teres. If you remember from your muscular lecture, I mentioned that teres means round. And this is remnant of the fetal umbilical vein. So we just see it along the free edge of that falciform ligament. More gross anatomy on the liver. We talked about the lesser omentum in class, and this is attaching the lesser curvature of the stomach. So if you could imagine, let me see if I could draw this in. Okay, here's our lesser curvature of the stomach in here. So this would be like my stomach, and this would be way over. Let me redraw this. And then the esophagus would go up. Not the best drawing here. Okay, there's our stomach. So this lesser curvature contains our lesser omentum all in this area, and then it attaches to the liver. Okay, there's your lesser omentum. Now the hepatic artery and vein. Is there a way I can erase this? There isn't. Okay, anyway. Nope, there isn't. Okay, so hepatic artery and vein is going to enter the liver at the porta hepatis. The porta hepatis is found at the hilus of the liver. So think of that hilus in your lung that you learned in our last chapter. It was an area or a location where structures are going to enter and exit. And that's exactly what it is here on the liver. Structures are entering and exiting here. So we have our hepatic artery and our hepatic vein. Really the hepatic portal vein here. Our hepatic veins are going to exit the liver and enter the inferior vena cava. It's a little bit different. And then we have our duct system. Our common, do I have a highlight here? Our common hepatic duct will leave the liver through this porta hepatis in the hilus of the liver and continue on down to meet our cystic duct. Now the cystic duct is this portion right here. It's draining the gallbladder. So we get bile that's made from the liver moving on down at the common hepatic duct, and then we have the cystic duct joining it. At that point, when it joins the common hepatic duct, it's going to become the common bile duct. Okay, so you can see this in the image below here, even though I've kind of drawn all over this and can't erase it. I'm going to see if I could just, oh, that was nice. Okay. So here we have our hepatic ducts. We have a right hepatic duct and a left hepatic duct, and these are going to join together to create that common hepatic duct. That joins the cystic duct, creating the common bile duct. Now this will be later joined by our pancreatic duct, which will all secrete its juices into the duodenum. So we'll secrete bile from this upper portion, the common bile duct, and pancreatic juices from the pancreas. Get all my highlights out here. Okay, there we go. Now when we look at a microscopic view of our liver, we're going to see some liver lobules. And you can see these are hexagonal structural and functional units of the liver. They have plates of our hepatocytes or liver cells that are going to filter and process that nutrient-rich blood. Remember, when we think back to, let me just go back since okay, I have a slide on it, so let me go back. Okay, here we go. We're just going to review real quick again. When we have our oxygenated blood go to our organs, okay, so whatever it is, the intestines, the colon, pancreas, spleen, stomach, that tissue is using up the oxygen, but we are gaining the nutrients that is being broken down by these organs and put into the veins here. So now we have deoxygenated blood and also nutrient-rich blood all moving into the portal vein and into the liver. So let me just go back to the slide that we were on. Okay. So where was I going with this? Okay, so it's filtering that nutrient-rich blood. And then we have a central vein at the center of each of these lobules. And when we look at the corners of these hexagonal lobules, we're going to see that each of them will have a portal triad. That portal triad will contain a branch from the hepatic artery, which will supply oxygenated blood to our liver. We'll also have a branch of that hepatic portal vein, which has the nutrient-rich blood I was talking about from our intestines. And lastly, we will have a bile duct, and this is receiving bile from our bile canaliculi. Remember, canaliculi are just small canals. We'll also get liver sinusoids that are created, these bluish lines over here. And I have it highlighted in orange, indicating it for you. And these are going to be leaky capillaries between our hepatic plates. And so our blood from our portal system coming in here within those triads, and also our hepatic artery proper bringing in the oxygenated blood will percolate from these regions over and into the sinuses down in here, and eventually will lead to our central vein, which will hook up to our hepatic veins and to our inferior vena cava to go back to the heart. Now within our liver, let me see if I have a picture on this. I don't think I do. We are also going to have special macrophages. These are resident macrophages within the liver. So we're going to call them hepatic macrophages, satellite macrophage, stellate macrophages, or they're also referred to as the Kupffer cells. And these live within these liver sinusoids to help remove any debris and old red blood cells. Now, our functions of our hepatocytes, our liver cells, they produce almost 900 milliliters of bile a day. They're also going to process our blood-borne nutrients, so they'll store glucose in the form of glycogen. Glycogen is just a long chain of a bunch of glucose molecules together. And they'll also make plasma cells like albumin. They'll store fat-soluble vitamins, so vitamins K, A, D, and E. And they'll also perform detoxification. And they could do this by converting ammonia into urea. Our hepatocytes, as far as a cell perspective, are also going to have increased rough and smooth ER, Golgi apparatus, peroxisomes, because they're detoxifying the fluid that's coming in, and also mitochondria. So now let's talk about the bile that it is creating. It's a yellowish green color, so I actually posted a picture here of what bile looks like, and it is a basic solution, so an alkaline solution. It's going to contain bile salts, which is represented here, and basically this is a derivative of cholesterol that will help to emulsify fat and absorb it. We'll also have bilirubin within our bile, and this is a yellowish brown pigment that is formed from heme when we break down the hemoglobin within our red blood cells. Bacteria is going to break down in our intestines to stercobilin, and that is going to give its brown color to the feces. So you can see the whole path here. Here's our bilirubin, the breakdown of heme moving in the form of bile, gets to our colon, and then will break down into stercobilin and create our stool or feces. We'll also have within the bile cholesterol, triglycerides, phospholipids, and electrolytes. Now let's talk about some hemostatic imbalances of the liver. Some of you may have heard of liver cirrhosis. We did talk about this in Biologists. This is a progressive and chronic inflammation from chronic hepatitis or alcoholism. So in the lab, you will see a healthy liver, and if you push on it, it's not spongy like a lung, but it has a little give to it. Whereas a liver that has cirrhosis, it's fibrous, it's rough, it's bumpy, it feels like a rock. So what is happening is we're having the healthy hepatocytes, healthy liver tissue, turning more fibrous and into scar tissue, and also becoming more fatty. And this can lead to portal hypertension. Liver transplants are going to be successful, but livers are scarce, and they can regenerate to its full size in 6 to 12 months after 80% of it has been removed. So that's promising. As far as our gallbladders, these are going to be thin-walled muscular sacs on our ventral surface of the liver, although ventral meaning anterior. So when you look in a cadaver, most of them you'll be able to find a little bit more easily if you look at the inferior surface. So here's our right lobe of our liver. This is on the right side of the body, and then left lobe would be closer to the left side of the body. Here's our gallbladder located near the ligamentum teres, and sitting next to the gallbladder here would be our quadrate lobe as well. What does our gallbladder do? It's going to store and concentrate bile by absorbing water and ions. It can also contain a honeycomb look to it, and that is going to allow it to expand as it fills up. And our muscular contractions of our gallbladder will help move bile into the cystic duct, which will flow down into the common bile duct and into the duodenum. And here we have a clinical correlation of gallstones or biliary calculi. So these are going to be examples of our gallstones that we can find in a gallbladder. This is caused by too much cholesterol and too few bile salts. If you remember from our previous slide here, our bile salts are going to fuse to a fat globule and then help to break it down or emulsify it. So let's go back. There we go. So when this happens, there we go. When this happens, we are stopping the flow of the bile from the gallbladder down into the duct system. So we can have our gallbladder become very painful when it's contracting against these sharp crystals. We can also have obstructive jaundice, and this will be a blockage that causes our bile salts and the pigments to build up in our blood, causing the yellow appearance to our skin or the sclera of the eye. Jaundice can also be caused by liver failure because we are putting too much of the bilirubin into our blood. The treatment for gallstones is going to be taking crystal dissolving drugs. We can use ultrasound vibrations through lithotripsy. So what it does is it sends those vibrations and causes these stones to break into smaller pieces for it to move through the ducts. There's also laser vaporization or surgery to have them removed. Moving on to the pancreas. Like I said, this pancreas is going to be retroperitoneal, and you can find it right behind the stomach, and it's deep to that greater curvature. So if you lift that up, you just find that pancreas hiding right in there. We have the head that is encircled by the duodenum, so you don't see it too well here, but the duodenum would just hug the head here. This is the body that transverses the vertebrae, and then the tail is going to point to the spleen, which isn't really pictured here. It's exocrine function. Now, when I say that, the term exocrine should be linked to the word duct. Exocrine and duct. Anytime we talk about an exocrine gland, that means it has a duct. Salivary glands are exocrine glands because they have ducts that secrete saliva. So our pancreas is also an exocrine gland. It's going to produce pancreatic juices that will flow into the duodenum. And it does this through acini, which, hold on, I think I have a picture of this, but forgot I put a cadaveric picture for you here to show you the pancreas. Here's the head of the pancreas being cradled by the duodenum. This is the duodenum. The body of the pancreas and the tail is in teal here, pointing to the spleen. All right, now here is our acini, and this is going to be a cluster of our secretory cells that produce zymogen granules that'll contain proenzymes to help break down food. Our ducts are going to all move into our duodenum here via this main pancreatic duct that I kept mentioning earlier. We do have smaller duct cells that will produce water and bicarbonate that'll go into this pancreatic duct as well. Now the pancreas is also an endocrine gland. So it's an exocrine gland and an endocrine gland. So that doesn't happen too often in our body. And when you hear the term endocrine, I want you to think hormones. So the hormone that it secretes is going to be insulin and glucagon, and it does this through the pancreatic islet cells. So what is our pancreatic juice made up of? It's watery and basic. So we have the pH of about 8 to neutralize the acidic chyme that we had within the stomach. And we're also going to have electrolytes and primarily the bicarbonate ion and digestive enzymes like our proteases for our proteins to help break them down. And it's secreted in an inactive form to prevent self-digestion of the pancreas. Amylase, we've already mentioned this, is secreted within our saliva, will help to break down carbohydrates. Our lipases we mentioned earlier as well. This is going to digest our lipids, and our nucleases, these are going to help to digest nucleic acids that we're typically getting from animal products. Proteases we mentioned are going to break down proteins. They're going to be activated once they move into the duodenum. We have an enzyme called the enteropeptidase, and this enzyme binds to the plasma membrane of our duodenal epithelium cells lined here in the mucosa, and it'll activate the pancreatic protease trypsinogen, which is the inactive form, into trypsin. So this is kind of a schematic here. Once trypsin is activated, then it goes back and it activates more trypsinogen. It's going to activate our procarboxypeptidase to our active form, the carboxypeptidase. We are also going to have trypsin move and take the inactive form of chymotrypsinogen into our chymotrypsin. Those are a mouthful. So I'm not too picky about you guys knowing these different proteases, but just know that these are going to help to break down proteins. All right, our bile duct and pancreatic ducts are coming together let's just review this here. We got our left and right hepatic ducts turning into our common hepatic duct, joining our cystic duct from the gallbladder, and when that happens, it becomes the common bile duct, and it's joining this main pancreatic duct from the pancreas to the duodenum. When it fuses together, this is now called the hepatopancreatic ampulla. So we're getting bile and pancreatic juices in the duodenum. This ampulla has a volcano-shaped major duodenal papilla. Now, sometimes we will also have another minor duodenal papilla, but it's only in a certain percentage of our population. Within the papilla, we're going to have a sphincter located there, actually just outside to it. This sphincter, like we said, when it contracts, it's going to close up the lumen of a tube. So it's not going to allow the bile and the pancreatic juices to move into the duodenum. So it'll kind of control that. We also have our, forgot to highlight these, our accessory pancreatic duct. So you can see this branches from the main pancreatic duct, and this will empty directly into the duodenum instead of joining with the common bile duct. To regulate bile and pancreatic secretions, they're going to be stimulated by neural and hormonal control. As far as our hormonal control, we have the cholecystokinin or CCK and secretin. I mentioned their functions a little bit earlier, and you can also look at the table in your book if you need to review them. Okay. Bile secretion is going to be increased when we have these three different things going on. We have our enterohepatic circulation returning large amounts of our bile salt. We have secretin from our intestinal cells exposed to our hydrochloric acid and fatty chyme that'll stimulate the gallbladder to release bile into the duodenum. This is going backward and down into the duodenum. And we also have that hepatopancreatic sphincter closing up. And that's going to be it for part 1. Now we will start part 2. So now we're going to start our part 2 lecture of the digestive system in chapter 23. First, we'll start with the gross anatomy of the small intestines. So our small intestines is really going to digest and grab the nutrients from our food and absorb it into our blood. So we have 3 different parts of our small intestines. We have the duodenum. Oh, I thought I had a picture showing you this, but I think it's on the next slide. We have the duodenum, which would be that first portion off of the stomach. The jejunum, which most of that is going to be located in the upper left quadrant. And then we have the ileum, which most of the ileum will be located in the lower right quadrant. This will attach posteriorly to the mesentery and join our large intestines at the ileocecal valve. Makes sense because that first part, ileo, is referring to the ileum, and cecal is referring to the cecum of the large intestines, which we'll learn about a little bit later in this lecture. So, what supplies the blood to our small intestines? You already know this. It's the superior mesenteric artery, and that comes off of the aorta, and you can see all these branches going to our small intestines here, as well as that first part of the large intestines. Now, we drain our blood from our intestines into the superior mesenteric vein. Remember, this is deoxygenated blood, but it is nutrient rich because we're pulling it from the intestines where we've gained the nutrients from the food. And so we are going to move on up and put this in our hepatic portal vein and finally into the liver. To give nerve supply to our small intestines, we have our parasympathetic nervous system, the rest and digest response through our vagus nerve. So that's going to come off of our brain. This is cranial nerve number 10, and it just moves on down and supplies our small intestines. Now our sympathetic innervation, which really stops the digestive process, is through our thoracic splanchnic nerves. We're going to talk about splanchnic nerves in Anatomy 3 when we discuss the autonomic nervous system, and I believe that's chapter 14. But basically we have branches coming off of our thoracic spinal nerve. We will synapse at a ganglion here, which a ganglion is just a location of cell bodies outside of the central nervous system. And from there, we will go down a nerve path to supply the intestines. We have 3 major modifications within our small intestines that's going to help us with absorption and to increase the surface area as well. And that is going to be the circular folds within the lumen. We also have villi, these little finger-like projections sticking up, and even smaller on these villi, on the mucosa, will be the microvilli. So let's talk about each one of these. Let's talk about the circular folds first. These are also called the plicae circularis. And these are going to be permanent folds. They're always there to force the chyme to slowly spiral through the lumen. And this gives more time for it, our intestines to pull the nutrients from our food. This is not the same as peristalsis, okay? Next we have the villi, those finger-like projections, and I'm just going to switch to a different view here where we're looking at it a little bit more in depth. So this is still a part of the mucosa. We see that simple columnar epithelium here. We see the lamina propria all in here. Excuse me, and within the finger-like portion, we're going to have some blood vessels and also our lymphatic vessel. Now this lymphatic vessel is a lymphatic capillary, also known as a lacteal, and this helps to absorb more of the fluid within the blood capillaries here. And then we have our microvilli. This is sitting on top of these simple columnar epithelia, and this is a nice little close-up view. We call this a brush border because it does. It looks like a brush. It has this fuzzy or grainy appearance when you're looking at it on a histology slide. And so we're going to have a bunch of enzymes, and I'm just going to give a general term for this so that you don't have to know all the different enzymes. We'll call them brush border enzymes, and they're used for our final carbohydrate and protein digestion. We also will have these little dips in between our villi, and these are called intestinal crypts. We will find tubular glands scattered in between these villi within these crypts. And just to give a little bit more clarity between the two functions of these, our villi are going to absorb the nutrients and also electrolytes, whereas our crypts, they're going to produce intestinal juice, which will have a watery mixture of mucus that can act as carrier fluid for our chyme. When we look at our intestinal wall, so here's our villi, those simple columnar epithelia will also have amongst them 5 major types of cells. First, we have our enterocytes, and these are going to be in the orange. This is making the bulk of our simple columnar epithelium. Our goblet cells, which are pictured in yellow here. Okay, you also saw that, well, it's represented here, I believe, by the purple, but usually when we see them on a histology slide, they have more of a whitish appearance because they're going to contain mucus, and they secrete that mucus into the lumen. And mucus is really important all throughout the digestive system because it has enzymes within it. It'll help to easily move the food through the digestive system. Okay, next we have our enteroendocrine cells. Remember, when we think endocrine, you want to think hormones. So these cells here are going to be a good source to secrete some of our hormones, as well as our enterogastrones. We have CCK, gastrin, serotonin, somatostatin, histamine and endorphins, and I'm recalling a test question on this, so make sure you recognize this. And these are directly released into our lamina propria here. So there's our enteroendocrine cell, all of these hormones secreted in this area at the lamina propria. Next we have our Paneth cells. Our Paneth cells are deep in the crypts. They're represented by the purple here, and they are specialized secretory cells that are going to fortify the small intestine's defenses. So they'll secrete antimicrobial agents like defensins and lysozyme that'll destroy bacteria. And the last type is going to be our stem cells, which are pictured in the red. And these are dividing to form a lineage of the other different cell types. You may see in this illustration we also have these cells in blue. These are known as transit amplifying cells. This is the transition from our stem cells into these other differentiated cells. Now let's talk about the intestinal wall as far as the lymphatic system. I mentioned a little bit earlier in our part 1 lecture that we have a lymphatic system in place with our digestive system, and that's MALT. And it stands for our mucosa-associated lymphoid tissue. It protects our intestines against pathogens invading and will include individual lymphoid follicles. So I have some pictured here. These are within the ileum pictured here. And Peyer's patches are just aggregated lymphoid nodules. These are located specifically in the lamina propria. Remember, lamina propria is still a part of our mucosa. And we have them most developed in the ileum, although they're found all throughout the small intestines. The reason we find them more in the ileum is because we have increased numbers of bacteria there. What else? Our lamina propria is also going to contain our plasma cells and secrete IgA. That's a specific antibody, and typically we're going to get that from breast milk and our placenta from when we were a fetus. And our submucosa is going to consist of areolar connective tissue, as it has all throughout our digestive system. And we'll also have duodenal glands located in our submucosa that will secrete an alkaline mucus to neutralize acidic chyme. That's more specifically in the duodenum, hence the term duodenal glands. So just a clinical correlation here, when somebody gets chemotherapy, this is targeting our rapidly dividing cells, such as our cancer cells. Now, the negative side effect to that is that it also targets these rapidly dividing gastrointestinal tract epithelia. The epithelium here is regenerating every 2 to 4 days. So this is the reason why many patients are undergoing chemotherapy and having symptoms like nausea, vomiting, and diarrhea. Let's go back to that intestinal juice that's created by our pancreas, liver, and gallbladder. This is all put into our duodenum. What it is we make about 1 to 2 liters daily in response to distension or any irritation to the mucosa. It is a major stimulus for production that is hypertonic or acidic chyme, and it is going to be more of an alkaline product. So it's more basic, and it is isotonic with our blood plasma. It's also going to be made up mostly of water, but it also will contain mucus. And again, that mucus is secreted by the duodenal glands and our goblet cells of the mucosa. Our chyme within the stomach is going to contain partially digested carbohydrates and also proteins, and then undigested fats. Remember, only about 15% of our fats will be digested in the stomach. So it could take 3 to 6 hours in the small intestines to absorb all the nutrients and most of the water that we've ingested. So where do we get all the enzymes to digest our macromolecules? Well, we get the bile from the liver and the gallbladder moving down our duct system. and also we get it from uh, our bicarbonate and other digestive enzymes from the pancreas, all filtering into the duodenum. And we have brush border enzymes that are going to bind to that plasma membrane of the lumen and perform the final digestion of our chyme. Chyme that enters our duodenum is going to usually be hypertonic. Let's review what that term hypertonic is, since I've mentioned it twice now. It means that we have an increased pressure or tone. And so our chyme delivery has to be slow in order to prevent osmotic loss of water from our blood. We also have our low pH of chyme in here that needs to be adjusted upward. That's why we want more of a basic solution within the duodenum. And the chyme has to be mixed with the bile and pancreatic juices to continue the digestion process. After a meal, we're going to have segmentation occur in the small intestines. Like we've mentioned before, it's going to help to mix and move the food, the chyme, toward our ileocecal valve, the last part of the small intestines. And the intensity is mixed with our long and short reflexes and also hormones. Remember that our parasympathetic nervous system increases this movement, and sympathetic is going to decrease it. Now, in between meals, our peristalsis is going to increase. So a wave will start distal to the previous wave. So here's our proximal wave. This is distal to it. This is distal to the one before that one. And so we'll have meal remnants, bacteria, and debris that'll all start moving toward the large intestines. And this, to complete a trip from the duodenum to the ileum, takes approximately 2 hours. And we do have a specific hormone called motilin that'll help to initiate this happen. And here is a nice little chart that gives us an overview of all of the digestive processes that take place in the different areas of our alimentary canal. So I'll let you look over this table. We've talked about these specifically. So next we're going to discuss the large intestines and just how our small intestines had some unique features. We're going to have some unique features of our large intestines. So the first one is going to be our tennae coli, and that is this light brown strip that you're seeing all throughout the large intestines. It's actually 3 bands that'll move through and around the large intestines, and this is made up of smooth muscle within our muscularis externa layer, that third layer of our digestive tract. We also have something called haustra. Haustra is plural, haustrum is singular, and these are pocket-like sacs that are caused by tone of the tennae coli. So they're kind of pulling on it and create this pocket-like sac to it. Next, we have our epiploic appendages. These are fat-filled pouches of our visceral peritoneum. So let's start learning the other parts of the large intestines. This first part I mentioned earlier is the cecum. It is a blind pouch. So here's our ileum entering the large intestines here at the ileocecal junction. So just below that area, up until that blind pouch, is the cecum. Now off of the cecum we have our vermiform appendix, vermiform meaning worm. So it does look like a little worm hanging off of there. And this has masses of lymphoid tissue here. They're actually doing a ton of research on the appendix and finding lots of different uses for them. If somebody has an issue with their ureter and their urinary system, they're actually removing the appendix and using it to replace a part of the ureter to bring urine from the kidney down to the urinary bladder. But anyway, this is a part of our MALT within our lymphoid system, lymphatic system, which works with our immune system as well. It is a bacterial storehouse that is capable of recolonizing the gut when necessary, And it also has a twisted shape that makes it susceptible to blockages. So that's why we may have appendicitis from time to time. Well, we only get it once really, but anyway, some people get it. The rest of the colon, we have our ascending colon, so this is just moving on up the right side of our abdominal cavity to the level of the right kidney, but it ends at our right colic or hepatic flexure. The reason it's called the hepatic flexure is because we have the liver sitting right there. Then our transverse colon is moving across from the right side to our left side of the abdomen, and we have another turn here at this orange arrow. This is our left colic or splenic flexure because we have the spleen in this area. And then moving down the left side of our abdomen is going to be the descending colon. And lastly here we have our sigmoid colon, which has an S shape or a loop shaped portion that travels down to the pelvis. And usually this area is the most packed with feces before you eliminate it. And another part of the large intestines, one of the last parts, is going to be the rectum. Remember, rectus means straight, so this is a straight part of the colon. We have 3 rectal valves that'll stop the feces from being passed with gas out of the digestive tract. We also have this end portion here known as the anal canal. This truly is the last part of the large intestine. Some people like to call it its own organ, and I'm just relabeling these for you. There's our anal canal, and this will open to our external environment. So I want to take a closer look of this anal canal here because we have 2 sphincters we need to check out. First one is going to be this internal anal sphincter, and this is made up of smooth muscle. So it's not going to be voluntary. This is involuntary. Our external anal canal, this is the one we have control over to allow the feces to move through and out the body because it's made up of skeletal muscle. Now let's talk about the relationship of the large intestines to the peritoneum. Remember, the peritoneum is the serous membrane within the abdomen, and we are going to have our cecum, this portion down in here, our appendix, little worm guy sticking off, our rectum, all being retroperitoneal structures. We also have the ascending colon and descending colon being retroperitoneal as well. I should have listed that here. But anyway, all of these are basically retroperitoneal except for the transverse colon and the sigmoid colon. We have our intraperitoneal regions that are going to be anchored to the posterior abdominal wall by mesentery sheets called mesocolons. So let me show you what those look like. Here's our cadaver. And like I said, when you first cut a cadaver open, you're going to see this big fatty apron sitting on the intestine. So that's what all this yellow gunk is. It's the greater omentum. And that again will extend off of the greater curvature of the stomach and cover the intestines. So let's move on to the next photo so I can show you a little bit more here. Let's review. Here's our lesser omentum attaching from that lesser curvature of the stomach to our liver. Okay, there's our greater omentum now cut away. So now we're going to look at another photo showing you the mesocolons. So first we have our transverse mesocolon. This of course is attaching the transverse colon to the posterior abdominal wall. And we have our sigmoid mesocolon that attaches the sigmoid colon to the posterior abdominal wall as well. And this is just another view of the lesser omentum, the greater omentum, and our mesocolons as well. Well, really we only see the transverse mesocolon because the sigmoid mesocolon would be found more laterally. When we look at the microscopic anatomy of our large intestines, we're going to see that we do have that simple columnar epithelium here with our microvilli, our brush border, and we're going to have a little bit more of a thicker mucosa here, except in our anal canal, which is pictured down in here, where we'll have that stratified squamous epithelium to withstand abrasion once again. So we won't have the circular folds or the villi that our small intestines have. We also won't have any digestive secretions like bile or pancreatic juice. And we will have an abundance of deep crypts. So here's another view of the colon. We have our simple columnar epithelium, and then moving on down, here are those deep crypts we were talking about. We have lots of goblet cells here that are going to secrete mucus. So all of these that look whitish, like white ovals, those are all goblet cells. So huge difference between this and the small intestines. Let's just note our bacterial flora. You guys will be talking about this in microbiology if you haven't already. But what bacterial flora is healthy bacteria within our gut. And we have over 1,000 different types of bacteria. And what we want to do is have our cells outnumber the bad cells, so that we create kind of some competition between the bacteria within our gut. We're also going to have vitamins made by these bacteria within our gut. So we're going to synthesize the B complex vitamins and some vitamin K needed by our liver to produce clotting factors. Residue that remains within our large intestines will be moved through between 12 and 24 hours. We won't have any food breaking down here except what our enteric bacteria starts to digest. We'll have vitamins which are created by the bacteria flora that we just mentioned, water and electrolytes, especially our sodium and chloride, that'll be reclaimed within our large intestines, and the other main function is to move the feces through and defecate it. Next, I want to mention a clinical correlation here. The term diverticulosis, you may have covered this in medical terminology, but we have the presence of diverticula. And basically these are little pouches, not like the Hostra pouches. These are smaller, and they're very common in all of our guts. Every one of us probably has diverticulosis. Just some of us have more than others. The most common area of these pouches are going to be in the sigmoid colon. Oh, I thought I had another picture here. And it affects half of people greater than 70 years of age. The more popcorn kernels or seeds that you eat, the more diverticula you'll have, because it starts to push on the wall and create the outpouches. Now, sometimes you can cause an inflammation in one of these pouches, and that's going to happen when we get some feces trapped in there, and then it starts to infect the tissue. And sometimes they may rupture and leak into our peritoneal cavity, which can be life-threatening. Defecation, basically moving the feces out of the rectum. So we have distension that's going to initiate the spinal defecation reflex, and we'll use our parasympathetic nervous system signals to stimulate the contraction of our sigmoid colon and also our rectum. And we'll relax the internal sphincter, and we have conscious control over our external anal sphincter. So we will, from the brain, move through the spinal cord, through our motor nerve, and tell the sphincter to to relax and allow the feces to move on out. Next, we're going to talk about the mechanisms of digestion and absorption. So chemical digestion is breaking down the foods we ingest by catabolic reactions. Remember, catabolic, you want to think to cut. We're breaking down the molecules into their chemical building blocks by enzymes. So I've listed for you how this happens, we have our larger units here, like our proteins get broken down into amino acids. Our polysaccharides, our carbs, gets broken down into sugars. Fats, lipids, and membranes turn into fatty acids, and nucleic acids turn into nucleotides. And we need them small enough to absorb them across the wall of our small intestines into our blood. So our absorption, like I said, is moving nutrients, those smaller building blocks, from the lumen of our gut into our capillaries. And our lipid molecules can be absorbed passively just through the membrane, but our other polar molecules, like our amino acids, need to be absorbed by active transport. And most of our nutrients are going to be absorbed before the chyme reaches the ileum. So let's talk about the digestion of each one of these macromolecules. First, we have the digestion of our carbs, and we will only be able to absorb monosaccharides. If you remember from biology, monosaccharides is just basically one sugar molecule: glucose, fructose, galactose. So we take our starch and our disaccharides and break it down into our oligosaccharides and disaccharides. So we have our disaccharides listed twice. That's fine, because some disaccharides are going to continue down to a different level. They can't be broken down by these amylases into our next set. So we will have our brush border enzymes working to break the oligosaccharides and disaccharides into lactose, maltose, and sucrose, which are more forms of disaccharides. And then from there, we'll further break them down into our single sugars, our monosaccharides. And that's going to be the lactose, glucose, and fructose. So let's talk about the steps of breaking down these carbs. So first we have our pancreatic amylase that's going to break down the starch and glycogen that escaped our salivary amylase into oligodendrocytes. And disaccharides, the brush border enzymes, are going to further break down these disaccharides and then into our monosaccharides, which are going to be represented by these orange and green molecules. So these monosaccharides are then going to be transported across the apical membrane. Remember, apical means that surface that is facing into the lumen, the top of the cell, and it absorbs through the cell and down to be pumped across the basolateral membrane by facilitated diffusion. Came to the wrong slide. There we go. So when it comes to the digestion of proteins, we're not only talking about the proteins that we consume within our diet, but it's also the digestive enzymes and the proteins that we get from breaking down our mucosal cells every 2 to 4 days. So our proteins are first going to be broken down into large polypeptides, then small polypeptides, small peptides, and then finally into our amino acid monomers. Remember, we have 20 different amino acids, and some are going to be dipeptides or tripeptides, basically 2 amino acids put together or 3 amino acids put together. And digestion for proteins starts in the stomach when we take pepsinogen and add it to our hydrochloric acid and turn it into its active form, pepsin. And that happens at the level of our pH 1.5 to 2.5. Once we move that pepsin into the duodenum, it becomes inactivated because now we have a higher pH level and it can't withstand that pH. Okay, so here is an illustration of our protein being broken down. So we get our amino acid chain here. So this is like a peptide. And we're going to use our pancreatic proteases, trypsin and chymotrypsin, to cleave the proteins into smaller peptides, while our carboxypeptidase is going to take off one amino acid at a time. So we're going to end up with these individual amino acids here. Our brush border enzymes are then going to break any oligopeptides and dipeptides into smaller amino acids too that we haven't taken care of previously. These amino acids are then moved through our pump again, this secondary active transport carrier, okay, through the cell, and then we'll exit through that basolateral membrane through facilitated diffusion once again, and then move into our blood. For the digestion of our lipids, we keep talking about how we emulsify our fats. So the emulsification status is when we have our triglycerides and their breakdown products that are insoluble in water. Now we need to pre-treat these with our bile salts that we gain within our bile to take that large fat globule and break it down into the smaller ones. Our digestion of lipids will consist of our pancreatic lipases, that'll break down the fat into a monoglyceride and 2 free fatty acids. As a result, we're going to get the formation of micelles, and these are going to be the products from digestion that become coated with bile salts and lecithin. Not lastly, but fourthly, we have diffusion, and this is when our lipid products leave the micelles and cross our epithelial membrane through diffusion. So we'll look at an illustration of this following this slide here. Next, we're going to get a chylomicron formation, and this is when we get our lipid products converted back into triglycerides and are packaged with lecithin and our lipoproteins to form a chylomicron. Then we're going to transport this chylomicron through exocytosis from the basolateral side of our cell and enter our lymphatic lacteal. Eventually, through our lymphatic system, this is emptied back into our venous system at our thoracic duct. The thoracic duct is a part of our lymphatic system. It drains the majority of our excess fluid of our body. Once in the blood in our venous system, the chylomicrons are going to be broken into free fatty acids and glycerol by our lipoprotein lipase so that they can be used by our cells. And we get our short chain fatty acids that can diffuse directly into the blood. So here's a schematic of this going on. We have our fat globule. Remember, we need our bile salts that comes from our bile that'll break off this large fat globule into the smaller ones. We'll get our pancreatic lipase to hydrolyze the triglycerides, giving us our monoglycerides and our free fatty acids. Then we get our micelle formation occurring eventually into breaking down into our fatty acids and monoglycerides diffusing through our epithelial cell on that basolateral side, and we get the formation of our chylomicron. Our fatty acids and monoglycerides are going to recombine and be packaged with other fatty substances and proteins to form our chylomicrons, and that moves into the lacteal, which is a part of our lymphatic system once again. So we're transporting from this lacteal throughout our lymphatic system and back into our venous system, excuse me, to be circulated in our blood. Okay, and then we have our digestion of our nucleic acids. So the nuclei of our ingested cells in our food contain DNA and RNA. So we use our nucleases to break them down. We take the nucleases from our pancreas. That's going to hydrolyze into a nucleic acid and nucleotide monomers. If you remember, those group of brush border enzymes are going to break the nucleotides into a free nitrogenous base, a pentose sugar, so deoxyribose or ribose, and also our phosphate ions. The breakdown of these products are going to be actively transported by special carriers in our epithelium of our villi. As far as our vitamin absorption, within our small intestines, we have our fat-soluble vitamins A, D, E, and K. I always remember the word CADE, K-A-D-E, to remember my fat-soluble ones. So I'll put that up here. Not the best handwriting, but that's okay. And these are carried by all the micelles, and they are going to diffuse into our absorptive cells. So our water-soluble vitamins will be vitamin C and also our B vitamins, that B complex. And they're going to be absorbed by diffusion or our passive or active transporters, so a number of different ways. That special vitamin B12 that we talked about in part 1, this is going to bind with the intrinsic factor and is going to be absorbed by endocytosis. In our large intestines, vitamin K and some of our B vitamins that were created from our bacteria flora are going to be absorbed there. Nope, went ahead too far. We have absorption of our electrolytes. I'm not going to be too picky about each of these statements that I've listed here. Just know that most of the ions are going to be transported actively along the length of the small intestines. More specifically, the most important ones, iron and calcium, are absorbed in the duodenum. Sodium absorption is coupled with our active absorption of glucose and amino acids. Chlorine is going to be transported actively as well. Potassium will diffuse in response to osmotic gradients, lost if water absorption is poor. We're going to talk about water absorption next. Usually the amount in the intestines is the amount that's absorbed. And our iron and calcium absorption is related to the need. So our ion levels are going to be stored in our mucosa cells with ferritin, and when needed, we'll have it transported in blood by transferrin. And calcium absorption is going to be regulated by vitamin D and parathyroid hormone. When it comes to our absorption of water, we have 9 liters of water, most from our gastrointestinal tract secretions that'll enter our small intestines. 95% of our water is absorbed in the small intestines through osmosis, and the rest is absorbed in our large intestines. So we have a net osmosis that'll occur if concentration gradient is established by active transport solutes, and water uptake is coupled with solute uptake. So the last thing I want to cover is some developmental aspects of our digestive system. We have a primitive gut here, so you can see it developing right in here, this light pinkish color all the way there. So it really is one tube when we are first starting to grow. And that primitive gut is an epithelial lining that's formed from the endoderm, and the rest of the GI tract comes from the mesoderm. So our oral membrane, this beginning portion in here, is going to become the mouth eventually, and will fuse to the scrotum, which I don't think is pictured here specifically. The cochleal membrane will become the anus, so that's going to be more toward this bottom portion where it says the cloacal membrane. And by week 5, our alimentary canal is continuous from the mouth to the anus. So you can see this is really starting to look a little bit more like a human or a tadpole, but you can see our developing gut and our accessory organs like the liver, the gallbladder, and the pancreas as well. So after all that is kind of developed, we are truly getting our nutrition not from ingesting food ourselves as a fetus, that wouldn't make sense, but we are gaining nutrition from our mother through the placenta. And then our GI tract is stimulated to mature by the amniotic fluid that we swallow in utero. And then once the baby is born, The newborn has a rooting reflex that's going to help the infant to find the nipple, and a suckling reflex that'll help it swallow the milk. And newborns typically will double in birth weight in 6 months, and will have an adult diet by 2 years. So I'm almost there with my son. Well, not almost at 2 years, he's almost 15 months, but he is basically eating adult meals. Anyway, we can also have things happen like gastroenteritis, inflammation of the gastrointestinal tract, and cholecystitis, which would mean we have ulcers or problems in middle age. And then as we get even older, we're going to start to have our gastrointestinal tract declining as we move up towards 60, 70, 80 years of age. We start producing less gastric juices, absorption is less efficient, peristalsis isn't going to be as efficient, and our bowel movements start to decline as well. So that is it for chapter 23. As always, if you have any questions, please feel free to contact me or come to tutoring on Tuesday in the Learning Commons between 10 and 12.